Okay, so today we're going to begin our lab in histology. And so histology is the study of tissues. So before we get to organ systems and individual organs, to understand how those work, we have to understand tissue structure and function because an organ is made up of different types of tissues. And so we really have to understand tissues before we move on. So what we're going to do in lab is you are actually going to identify the tissues on histology slides or microscope slides, so using the microscope. So all tissues in your body, every single tissue that you have, can be grouped under or classified as one of the four major tissues: epithelial, connective, muscle, or nervous. So on the exam, when I'm asking you to identify tissue slides, I'll ask you to tell me the specific type of tissue and the general type of tissue. The general would be one of these four types, okay? So one of these four types: epithelial, connective, muscle, or nervous. These are the general types of tissues. And for this video, we're just going to focus on epithelium for today. So epithelium, when you think epithelium, you want to think linings or coverings. So linings of body cavities, linings of tubules, the epidermis, it's a lining of your skin, okay? So it covers our body surfaces, lines internal organs, body cavities, and forms glands. So the main functions are going to be, and I'll talk about the specific functions as we go through each one, but the broad functions are protection, secretion, and absorption. So how we classify, so when we're looking at tissues, how do we know that we group all this specific type of tissue as an epithelial type, right? It's because all epithelial types share these general characteristics. And so general characteristics: a free apical surface, so free apical surface, and a basement membrane. What I mean by that is, if you look at this image right here, this is showing you simple squamous epithelium. But up top, so these would be our epithelial cells, and above it would be our free apical space, so open to the outside. Doesn't have to be open to the outside environment, but open to the outside like a tubule, facing the lumen of a tubule or the lumen of your intestines. So there's a free apical space, right, open to the outside. And then below the epithelial cells would be a basement membrane. A basement membrane is a very thin layer of connective tissue which all of these epithelial cells are anchored to. And then that basement membrane would be anchored to connective tissue below. So all epithelium is going to have some sort of connective tissue below it and a free apical space above it because it's lining or it's a covering. Tightly packed cells, so back to this picture. Epithelium, because they're forming linings or coverings, that means these cells have to be joined by cellular junctions. They're not just floating around. A covering or a lining isn't really good at doing its job of being a lining or covering if the cells aren't tightly packed together, joined by cellular junctions, primarily tight junctions or desmosomes. So tight junctions form an impermeable barrier so that anything that needs to get through that lining or covering has to pass through the cells. It can't squeak through the in between the cells because we have these tight junctions like a zipper in between the cells that forms an impermeable barrier, which we find in the intestines because, of course, we don't want our food, we don't want if we think of hydrochloric acid going from the stomach into the intestines, we don't want all that hydrochloric acid to squeak through the cells in between them, right? So we want to form an impermeable barrier. Or desmosomes, which are really strong cellular junctions that resist shearing forces, which we find in the epithelial cells that make up the stratified squamous epithelium of our epidermis, right? So they're tightly packed together. That means that they're joined by cellular junctions. Lack blood vessels. So because they are tightly packed together and they're joined by cellular junctions, they are not vascularized. There's no room for blood vessels to go through those tissues. So they rely on diffusion of nutrients from so there are connective tissue will be vascularized. and to what extent we'll talk about when we get to the connective. But connective tissue, remember me saying that the basement membrane is anchored to connective tissue below. Well, in that connective tissue below, you'll find blood vessels. And so epithelium will rely on diffusion of nutrients from the blood vessels in the connective tissue below up to the epithelium above it, okay? And same thing, the waste that's generated from these cells will diffuse through the connective tissue and into the blood vessels of the connective tissue. So, but the epithelium itself is not vascularized. So lacks blood vessels. Some have apical specializations based on its function. So what that means is some epithelia you will find on that free apical space, you'll find microvilli or cilia. When you think microvilli, you think absorption. Okay, so microvilli is actually invaginations of the plasma membrane. If you increase the surface area of the plasma membrane with folds or invaginations, increased surface area means increased ability to absorb through that plasma membrane, right? So microvilli, absorption. Invaginations of the plasma membrane increases surface area. Cilia are apical specializations for motility. So they're attached to the outside of the cell and they beat in a wave-like pattern, and it's for movement of things through that lumen or apical space, okay? Classified according to shape and number of cells. So what that means is shape, we have three basic shapes: squamous, cuboidal, or columnar. So squamous is flattened, irregularly shaped cells. So very flattened, or squamous means squished, right? So squished cells, if you look, this is an example of simple squamous epithelium. Flattened, irregularly shaped cells, and the nuclei will be somewhat oval or elongated, okay, in the cells. Cuboidal, exactly what that sounds like, cube-like. So these cells would be roughly equal on all four sides, giving it a cube-like appearance. And then columnar, meaning they are taller than they are wide, and so they look like columns. So this is columnar. Number of cell layers. Simple means a single layer of cells. Stratified means multiple layers. So if you look up here, we have simple squamous, a single layer of flattened cells. Simple cuboidal, a single layer of cube-like cells. And if you look at the nuclei of simple cuboidal, they're very spherical and they're located somewhat in the middle of the cells. This would be simple columnar, so a single layer of tall column-shaped cells. And the nuclei will be somewhat oval-shaped, and a lot of times you'll see it closer towards the basement membrane. So this would be where the basement membrane would be located, and up here would be that free apical space. Stratified, so down here showing you stratified squamous epithelium. So stratified because there's multiple layers, but if you look up towards this free apical space, they're very flattened and squished, irregularly shaped, so stratified squamous. Here, so you will not have to identify stratified cuboidal, but it does exist in the human body, usually in very large glands. But for our purposes, our identification purposes, you'll only have to identify a simple cuboidal tissue type. Okay? But stratified cuboidal means you have multiple layers of cuboidal-shaped cells. Pseudostratified columnar. So break down the word. Pseudo means falsely, and then stratified means multiple layers. So this is a type of epithelium that appears to be stratified, but is actually a single layer of cells. So, and columnar because it is a taller cell. See how this, if you look at simple cuboidal, very short, cube-like cells, these give a columnar appearance. And because the nuclei are very jumbled, if we compare it to the simple columnar above, the nuclei are somewhat all in a row, okay? The nuclei are very jumbled because not every single cell is reaching the apical surface. If you look at this, compare the simple columnar, every single cell touches the basement membrane and every single cell touches the free apical space. Pseudostratified columnar, every single cell is touching the basement membrane, so it's a single layer of cells, but this cell, See, there's a few cells that aren't reaching all the way up to the free apical space, right? So it gives this jumbled appearance and it looks as if it's stratified, but in actuality, it's a single layer of cells. This is pseudostratified columnar epithelium. Transitional is a type of stratified epithelium, but we call it transitional because if you look at the layers as you move up from the basement membrane to the free apical space, it looks as if it transitions shapes. The shape changes. It's not really continuous throughout. So this is transitional epithelium. So these are all specific types of epithelium, right? So simple squamous is specific. General would be epithelium. So let's go through the types of epithelium that we have. So simple squamous epithelium, again, it's going to be a single layer of flattened cells. They're irregularly shaped. This would be as if, this is an image, a micrograph. If you were looking at simple squamous epithelium from above or below, it gives like a mosaic type appearance. How you'll see it on the microscope is actually this way. So this is a micrograph from the lungs. And so if you look, all of these pink cells, the pink is not actually the color of the cells in the human body. So in order to visualize tissues, we have to stain them. So there are lots of histological processes where you have to dehydrate and embed and then stain these tissues in order to actually see them on a microscope slide. So don't take those microscope slides for granted because a lot of work goes into making just one little tissue slide. So this particular lung slide was stained with an acidophilic stain, so the meaning it made the cells pink-ish, so a pink red. But you can tell even from a far out distance, so this would be a low magnification, this is a single layer of flattened cells, okay? So for the exam, you'll have to be able to identify a tissue, right? So you'll have to be able to tell me the specific. So if I have an arrow pointing here to this tissue, you would have to tell me the specific, which is simple squamous, and then you would have to tell me the general type of tissue, which would be epithelium, okay? So simple squamous is the specific, epithelium is the general. You also have to be able to tell me locations and functions of these tissues. And so for a simple squamous epithelium, locations, ideal locations, you find it in the lungs. You also find it in capillaries, which are the smallest blood vessels where gas exchange and nutrient exchange happens between the blood and the tissues of our body. So why would we find simple squamous epithelium in the lungs and in the capillaries? Well, a single layer of flattened cells, right, makes diffusion a lot faster. So diffusion, what increases the rate of diffusion? Well, increased temperature, the size of the molecules, right? The smaller the molecules, the faster, But distance also affects the rate of diffusion. So the shorter the distance, the faster the rate of diffusion. And if we go back to this image right here, if we're trying to get oxygen through, because remember these cells are connected by cellular junctions, which means that things can't squeak in between cells. Anything that wants to pass these layers, they have to pass through the cells themselves, right? So what would be faster? Oxygen trying to get through a single layer of flattened cells, through cuboidal, or through columnar, or through multiple layers? If you look, the distance is greater in all of these. This is the shortest distance. So in order to increase the rate of diffusion, you decrease the distance that those molecules have to diffuse across. So ideal locations, lungs, because we want oxygen and CO2 to go across that lung tissue very, very quickly. Capillaries, because we want nutrients and gases to diffuse from the blood vessels to the tissues and vice versa very, very quickly. Okay, so simple squamous epithelium. Anywhere where you want diffusion of nutrients and gases to happen very quickly. Here's another image, and you can really see that this is a single layer of flattened cells, simple squamous epithelium. And those darker areas in each cell, that would all be the nuclei. Okay, so those would be the nuclei, those darker staining circular structures within each cell. This is actually from the lung, and I know that because you see all these air spaces, right? This is actually where air would fill up when you inhale, okay? So these are the alveoli of the lungs, big air spaces. Now, you don't have to be able to tell me you don't have to be able to identify lung on a microscope slide yet, but you do have to be able to look at this slide, be able to notice that this is a single, if I have an arrow pointing here, notice that it's a single layer of flattened cells. You have to be able to tell me simple squamous for the specific and epithelium for the general, okay? Usually when I ask simple squamous epithelium, it's going to be from a lung slide, and you'll see all of these air spaces, okay? Now, if you want to test if you can find simple squamous on a more challenging slide, this is actually from the kidney, right? So this is the kidney. And when you're looking at the kidney and you're going on higher magnification, you'll see these little balls. These are glomeruli from the kidney. And this is a tuft of capillaries. There's this space. You'll see lining this is a single layer of flattened cells. That's simple squamous epithelium. So higher magnification. Here's that tuft of capillaries. Here's that space. And here, this is simple squamous epithelium. And you can really see that flattened, squished cell, another flattened, squished cell. This would be a nucleus of one cell, a nucleus of another, a nucleus of another. So a single layer of flattened cells. This is simple squamous epithelium in the kidney.